We cannot check if people are back, but we'll assume that. So we are very happy to hear Diala Abu Awad, who is Humboldt Fellow at Technical University of München. She will speak about the co-evolving genome. Thank you very much.
Thank you. So like everyone else, I'm going to thank the organizers because they really made it through all of the challenges that they had to face for this, to bring this meeting together. Today, I'm presenting not even a work in progress, it's a thought in progress, because this is something I have been thinking about for a few years, but I don't know how to approach it in a simple and biologically realistic and mathematically feasible way. So yeah, if you have any questions during the talk, please don't hesitate to interrupt me because I might be using some biology heavy vocabulary, and if you're not okay with that, just let me know. So we consider genomes as a fixed trait, which is a very necessary simplifying assumption when we're trying to study the evolution of species or populations when looking at specific traits. And also when we're building inference models that go back into the past for over, I don't know how many thousands, hundreds of thousands of generations, we cannot assume that the genome is dynamic and going all over the place. But the question that I've been wondering about is how much are we actually missing out on by not considering the genome as something that is evolving and co-evolving along with other population traits and what's the word, and properties. So just to give you an idea of what we know about genome sizes, here we have a collection of genome sizes in megabases as a function of the recombination rate that's scaled to the genome size in a way, and the genome size as a function of the mutation rate as a function of the genome size. And what we see, first of all, is that we have a very wide distribution of genome size. So we're going like from 10 to the power of 1 to 10 to the power of 5 megabase pairs. And this is a very large range, right? And it's spread between all species, with mostly unicellulars keeping smaller sized genomes, but nonetheless widely distributed. And we have plants that seem to have the higher genome sizes. So there seems to be, so there is definitely this very large distribution. And on top of that, it seems to be correlated with other genomic processes. So recombination rate, for example, seems to be negatively correlated with the genome size. So the smaller the genome, the higher the recombination rate when it's rescaled. And the mutation rate seems to be positively associated with the genome size. But this result here is to be taken with a grain of salt because measuring the mutation rate is a bit complicated. Now, these are actually average genome sizes. These are genome sizes of scaffolds that were built from sequencing a single individual usually. So they don't give the whole picture of what's happening. And what I'm showing you here is the distribution of genome sizes between, on the left here, so this is genome size for different sister species of snapping shrimp. And already here we can see that there is a large distribution of these genome sizes, despite the fact that all these species are very closely related. And it even goes beyond that. When we look at a single species, we look at different populations, even within different populations, we see that there's a distribution of the genome size and that not everybody will have the same genome size. Now, I'm talking specifically about genome sizes here because it's very difficult to get other data on genome evolution. This is one of the more accessible traits or values, even though not everyone agrees on what genome size is and how to measure it, but that's another problem. We'll pretend that we figured that out. And so even though this is not the ideal trait that we want to be looking at, we can ask ourselves, okay, what really makes the genome size evolve. So there are two general schools of thought. There's one that's Lynch presented in his book of 2010, which is a very comprehensive work worth reading, definitely, where it's a more of a neutral aspect to genome evolution. So genome size will simply increase due to drift, so genetic drift, which is stochastic processes that are just happening. So it can be due to duplication that just happened randomly, or recombination that inserted something. So just very random things happening. And any processes that decrease the genome size, there might be some light selection, which is due to the cost of replication. So if the genome gets too big, it takes more energy to to replicate it every single time, to duplicate the cells. And so there is some kind of avoidance of going to genomes that are too large. Or there's also the other school of thought for this decrease, which is just these very small random deletions that happen completely randomly. And so there's no selection at all. Then there's another school of thought where selection is actually a main driving force in genome size evolution. One of them is this whole school of thought that has to do with genome robustness, where they consider, so the authors that write these specific papers consider that genome size is a direct reflection of the number of functional genes present in an individual's haplotype, and that the more functional genes you have, the less likely a mutation that will make one of them inactive one of them, the less probable that this will have a strong effect on your fitness, basically. So it's kind of like drowning out selection just by sheer number. And then there's the other point of view, which sees large genome size as a maladaptation, because if you need to duplicate your genome really quickly every single time, it's going to reduce the speed of cell division, and this is problematic. In general, these schools of thought all seem to agree on one thing when we look at how these, what these hypotheses entail. It's that, larger effective population size, which is a measure of genetic drift, so how much stochasticity has an effect on population evolution. So larger effective population sizes should favor smaller genomes in general, because selection will be better. Now, we can ask ourselves, because of this relationship between the expected genome size and effective population size, can there be a link with genome size and life history? And this is because, I mean, life history, which is basically lifespan, or investment in reproduction, or reproductive strategies, all of these different things, they will affect the effective population size. And so how much of a role do these life history traits play? And will selection be an important factor in determining genome size, or will we always remain in this kind of nearly neutral selection? And I will mostly be talking about, or only be talking about, the case of self-fertilizing species, because this is, for those who know me, this is kind of my obsession. And I want to be completely clear, I will not be able to answer these questions. I can't answer them. And this is just an opening for discussion, because I don't have an answer to any of these. Now, just to introduce quick self-fertilization to those who don't know it. Basically, there's a little error here. Basically, self-fertilization implies a hermaphroditic individual is able to produce both male and female gametes, and to fertilize its ovules with its own sperm. And this is really self-fertilization. And as we can see intuitively from this little cartoon, if an individual self-fertilizes, it will not only decrease the genetic variation in its offspring compared to an individual that will outcross, but it will also increase homozygosity. Now, it's very important to note that self-fertilization should not be confused with asexuality or clonality. because recombination does make a big difference. So if an individual has this blue and this orange haplotype, if it self-fertilizes, there will be meiosis, there will be recombination, and it will be able to transmit a different combination of its initial haplotypes to its descendants. So you increase the mix of genetic combinations, basically, through this recombination. So it's not exactly asexuality, that changes things. Now, self-fertilization is actually very widespread. It exists in both hermaphroditic plants and animals. And we have a very large distribution. So here the number of animals, or the proportion of animals with a specific selfing rate, are in gray, and the plants are in white. And what we see, there are species that will not self-fertilize at all, and others that will have intermediate rates, and others that will predominantly self-fertilize. And today we've come to an estimation of about 15% of plant species that will predominantly self-fertilize. So self-fertilization has several advantages. One of them is the most classic ecological one, which is reproductive assurance. And that simply implies that an individual that cannot self-fertilize will never be able to colonize a new habitat on its own. There'll have to be several individuals that arrive at the same time. Otherwise, it won't work. So kind of like the viruses that we just saw. And a self-fertilizing individual, on the other hand, will arrive into a new environment. There's no one else, and that's completely fine. They'll be able to colonize. That's not an issue. Furthermore, because of the fact that individuals have these homozygote descendants, so descendants that have two exact copies of the same genes, if a beneficial version of a gene, a beneficial allele, comes through mutation, it'll go to fixation much faster. And in a complementary way, if there's a deleterious mutation that comes in through mutation, it will be removed very quickly. Now, there's another half advantage, not really an advantage, which is the automatic advantage of self-fertilization, which is basically an individual that self-fertilizes will transmit more copies to the next generation than one that simply outcrosses. And so what that means is that any allele that will increase self-fertilization will automatically increase in frequency, even if it's not selectively positive to do so. So this is why this is the automatic advantage. It's kind of like a selfish gene. So when we take these advantages and the automatic advantage all together, this basically leads to a situation where we have a unidirectional evolution to high selfing rates. So once self-fertilization evolves, it will continue to evolve to higher and higher rates, and then we can never come back. Now, since self-fertilization has all of these advantages, we can say, okay, that's not a problem, but it actually is, because self-fertilization will actually reduce the effective population size, which means that it will lower the genetic diversity and lower adaptive potential, and favor the fixation of mildly deleterious mutations. And this could lead to a mutational meltdown, which is basically the extinction of a population because of this repeated fixation of deleterious mutations. Now, this should potentially lead to higher extinction rates, and led to the I think I mixed up the slides there. So this should lead to higher extinction rates. So to test these, I'm just going to show you two very simple studies that examined the adaptive potential and the mutational meltdown potential of populations. So in this simple model by Glemin and Rochefort, what they did was they took a simple case, a simple locus under selection, where there was some genetic diversity due to a deleterious mutation in the population. And then there was an adaptive environmental change, and they followed the time it took for populations to adapt as a function of their selfing rate, and the probability of extinction before they adapted as a function of the selfing rate. And as you can see here, as we reach a very high selfing rate of 1, the time to adaptation suddenly increases. And we notice that this is also linked to a very high probability of extinction. So that means that, yes, self-fertilizing populations should not be able to adapt to changing environments, at least not very efficiently. They will probably go extinct before they do. Concerning the mutational meltdown, Here's another model which follows only deleterious mutations, so mutations that will decrease viability or the reproductive success. And we follow the evolution of the rate of self-fertilization, which of course evolves to 1 because of the automatic advantage and the purging. And we look at the percent of viable populations at the same time as a function of the effect of the mutations that are being introduced into the population. When the mutations are strongly deleterious, we have evolution to a very high selfing rate and all populations remain viable. However, when mutations are mildly deleterious, we see that there starts to be a point after which mutations start going to populations start going to mutational meltdown, and they all go extinct within 25,000 generations for this parameter set. So these two factors together have led to the hypothesis or the suggestion that self-fertilization is an evolutionary dead end. And this is due to the fact that it's unidirectional and the fact that they're not able to adapt as well, and there's a potential for the mutational meltdown. And this seems to be confirmed from phylogenetic evidence, where here, this is a very important paper from Goldberg and al., where they looked at the net diversification rate of self-compatible, so able to self-fertilize, and self-incompatible species, so not able to self-fertilize, within the Solanaceae family, so that's tomatoes, potatoes, tobacco. And what they found was that the net diversification rate, which is basically the speciation minus the extinction rate of species in this phylogeny, were actually negative, which means that selfing will go extinct more than it will actually than it will actually evolve. So this kind of supports this evolutionary dead end. But the problem is that this failure to adapt in this mutational meltdown from the theoretical body of work that exists, they would occur too fast for them to be observed on a phylogenetic scale. So just to give you an example, Arabidopsis thaliana diverged 5 million years ago from Nirata. So if it was going to have issues with adapting to a new environment, it would have already gone extinct. So there must be something else that's going on. So this kind of brings me to talk about the selfing syndrome. And this is kind of what brought me as well to this line of thinking about genome size. So when there's a transition, a successful transition from outcrossing to selfing, and there's a speciation event and a species that is long-lived, there are clearly changes that are observed and that seem always to be there when we compare sister species that self-fertilize and don't self-fertilize. So first of all, there's a change in reproductive investment. So that means that, for example, here we have this flower that's outcrossing, it evolves selfing. We see that in the outcrossing flower, the petals are larger, it's more attractive, the pollen production is here, it produces more pollen. And then when it evolves to selfing, the petals are not as large, it won't be attracting as many pollinators, for example. And with the organs producing the pollen, so the sperm basically, are much closer to the stigma, which will lead to the ovules. So basically, there's a reduction in effort to export pollen, and everything stays within the plant so it can reproduce with itself. Another example is in C. elegans, which is a nematode. What they found was that in hermaphroditic populations, the females, which are larger than the males, the females will actually invest resources in producing very strong pheromones to attract the males. And they completely lose this ability in the hermaphroditic populations where there are no more males. So they're saving the energy from producing this pheromone to do something else. Another thing that happens pretty often is a shift to an annual life history. Whereas, so when we have species that are initially perennial, when there's the mating system evolves to a selfing one, it's also accompanied by a shift to an annual life cycle. So the individual will no longer survive for several generations to reproduce. And last but not least, there is a decrease in genome size. Now, concerning these two, this change in reproductive investment does seem to fit in with this shift to an annual life history. And this can be shown in with a very nice model that, oops, the names are not there anymore, by Lesaf and Billard, which follows the evolution of the investment in reproduction as a function of the selfing rate. And what this investment reproduction is basically individuals will either invest in reproducing now or in surviving. So the more there's investment in reproduction, the less there is in survival. And so lifespan will decrease. And we generally see that there is an upward trend, even in this case where it's not very strong, but there's an upward trend in this investment in reproduction as the rate of self-fertilization increases. So this does seem easy to explain and plausible, because this is entailed by the fact that there are mutations that accumulate. And so it is more advantageous for selfers to reproduce immediately and avoid going into later life stages. Now, when we look at these two, the fact that there is a shift to an annual form of life history accompanied with a decrease in genome size, this reminds us a bit of the genome size as a maladaptive hypothesis, as being maladaptive hypothesis, where what's important is the speed of replication. So as we evolve to an annual life history, we will also decrease genome size so that we can reproduce faster. Now, what is causing what is not clear. And I don't know if we have a way to find out, but it would be worth exploring, definitely. Now, the last point is just simply looking at the decrease in genome size that accompanies self-fertilization. And this is unfortunately counterintuitive when we look at the hypotheses that I gave at the beginning, which is basically when we have the evolution of selfing, we decrease the effective population size, which means that we should have a larger genome size because we're not able to select for smaller genome sizes. And this is not at all what we're observing. So what exactly is happening? When we, oh, sorry, I had it all here. Sorry about that. So when we look at the basically the theory, what is predicting so far, the genome size should increase due to drift. If the effective population size is small, there's more drift. If the effective population size is too small as well, it's better to be able to, it's better to have robustness to mutations. Otherwise, the individuals will go faster into mutational meltdown. And so we can't really conclude from the existing theory what should happen to selfers, because on one hand, we have the evolution of annuality, which will help decrease the genome size. And on the other hand, because of drift, we should increase genome size. Now, what makes up the genome in general? So I think I prefer to put this here because I'm going to be talking about differences in genome components in Arabidopsis thaliana and lirata, which are two sister species, one that self-fertilizes and one that doesn't. Within the genome, we have functional and non-functional genes, and often duplications, among other things. There's non-coding DNA, which before used to be called junk DNA, but now we're considering more as being DNA that offers structural support. So it's not really clear what's happening there. And of course, the biggest culprit is transposable elements, which can greatly contribute to increasing genome size. Now, what exactly is lost when selfers end up with a smaller genome size compared to outcrossers? When we look at Arabidopsis lirata, this is a perennial outcrosser, so it lives for several generations. First of all, we see that when we look at its genome is much larger than Arabidopsis thaliana, which is an annual and a selfer. So here it's 207 megabases, and here it's 125. So thaliana lost a big chunk of the genome once there was the shift from outcrossing to selfing. Furthermore, when we look at the coding, the proportion of coding regions within the genome, we see that lirata has a lower proportion of coding genome. Like less of the genome is actually holds coding DNA, even though it actually has a higher number of genes. So here it has 32,000 and thaliana has 27,000. And then the last thing that we will be comparing is the proportion of transposable elements, where Lirata has a higher proportion of transposable elements and also a much higher number of transposable elements throughout its genome. And what's happening in Taliana now is that there are still hundreds of thousands of small deletions all over throughout the whole genome. And it seems that selection is still ongoing, although separation, they diverged 5 million years ago. So there's something actively happening within this genome to make it smaller and to maintain it smaller. Now, basically I'm sorry, I think I might have missed something here. So basically, when we look at genome size, it's not enough, right? Self-fertilization will also have effects on other genome properties. So, for example, we know from theory, and we also have seen this in data, that when there's self-fertilization, when self-fertilization evolves, there's a higher recombination rate, which means that there's a higher probability of deletions happening and also maybe insertions happening. And so there's more movement occurring throughout the genome. There's also a potential increase or decrease in the mutation rate. And that depends on the effective population size. So there are cases where self-fertilization will be so good at selecting against a high mutation rate evolving that it will keep a much lower mutation rate compared to its outcrossing counterparts. Whereas in other cases, if the effective population size is too small, drift will be so strong that, on the contrary, the mutation rate will increase and will actually reach unbelievable or like unrealistic predicted values. Now, there's also the possibility that self-fertilization, because of the changes in the genomic changes that are occurring, there may be an effect on robustness. So just to be clear on what robustness is again, robustness is basically if a new mutation arrives in the genome, it will have very little effect because the genome has ways to compensate for it. Now, for me, the results that I'm going to show right here are counterintuitive, and I'll explain why. So this was basically a study where they looked at the fitness of individuals with time. This was in Arabidopsis thaliana, so it's very selfing. And they measured the fitness as the number of seeds per fruit. And what they saw was, so they started the first generation here. This is the distribution of the number of seeds per fruit, with the mean here. After several generations of pure selfing, so they didn't let them outcross a lot, so they can accumulate mutations. What we see is that we have a distribution of fitness that is very different from what we started with. And a lot of the new lineages that have accumulated mutations actually have better fitnesses than what was in the original population. And so this had led the authors, Shaw and al. In 2000, to come to the conclusion that selfing populations may actually, first of all, have fewer deleterious mutations that appear, and end up having more beneficial mutations that appear within the genome. Now, there are several problems with this kind of approach. The first is that because these are mutation accumulation lines, that means if there are very strongly deleterious mutations, they will automatically be removed from the experiment, because that means the seeds will not germinate. So we will never see these individuals. So this biases the distribution here, because these are things that we cannot observe. And because of the selection, we may actually be favoring these traits here. So that's one issue with it. And the other issue is that the way I would expect robustness to evolve from the existing theory is that, on the contrary, because self-fertilization will end up fixing mildly deleterious mutations because of the effective drift, it would actually be, my hypothesis would be that self-fertilization would favor a more sensitive genome, which means that any new mutations would be so strongly deleterious that they would be immediately removed and not have the time to accumulate. And it seems that in populations of Arabidopsis thaliana, there are no signs of genome degradation that would be linked to the accumulation of deleterious mutations. So we can ask ourselves that question of whether it would be more robustness or sensitivity. So the question is open. If we have long-term persistence of selfing, could it be because we are making the assumptions in our models and in our approaches that mutations will always have the same effect as self-fertilization evolves? Is that why we're predicting that self-fertilizing populations will go extinct so quickly? Could it be that maybe during the course of evolution of self-fertilization, the shock of the change of the reproductive system, the shock that it has on the actual genome structure and on selection throughout the genome, what effect would that have on the effect on selection itself and on the effects of new mutations themselves that appear in these populations? And it's an open question. I'm willing to talk about it, and for you, if there are any points of discussion where I've made strong assumptions that I shouldn't have made, I'm more than happy to change my mind if I have to. And with that, if you have any questions, I was faster than I thought I would be.
Thank you very much. Do somebody have a first question? I can begin with a very naive one.
Please go ahead.
So you said self-fertilization is not the same as sexual reproduction.
Asexual reproduction.
Yeah, exactly. So in fact, if I'm not, maybe I will say a lot of mistakes, but for sexually reproducing species, you have this diploid structure. So somehow, to compare genome size, you have a factor of 2 in the total number of bases. And
okay, yeah.
So it's not very. So I just want to compare your self-fertilizing species with sexually reproducing species, which would have actually twice the number of bases, right?
Yeah, but that in itself is not the problem here. So basically, you can have asexual species that are also diploid, so that have 2 copies of the genome. This also exists. So, so asexual is reproduction when there's a pure copy from the maternal genome, and it's given to the offspring as is. So it's the same process as when your own cells divide.
Even in the same, okay.
So asexuality is clonality.
Yes.
So it's the creation of clones. When you have clones, there's no recombination. So even though it would seem like cloning or self-fertilizing are the same thing, they're not, simply because when you're self-fertilizing, there is meiosis, and so you will end up recombining the haplotypes.
So it's also the case in your self-fertilizing species.
It is also the case in self-fertilization, exactly.
So it's exactly the same. No, it's not exactly the same advantages with respect to selection, because you don't need a partner to mate with.
It's not only you don't need a partner to mate with, it is also because of the fact that you're the only parent giving to the next generation.
Mm-hmm. Yeah.
If you have a mutation that's a beneficial mutation, you're sure to transmit it to the next generation. When you're reproducing with someone else, so let's say we're comparing this guy here reproducing here, and then reproducing together. Let's say the orange allele here, the orange haplotype, is very beneficial. If this individual outcrosses, only half of its descendants, well, actually, yeah, it'll only transmit 2 copies of this very beneficial allele to the next generation. But if it reproduces with itself, it's already transmitted 3 copies to the next generation. So this is where self-fertilization brings an advantage, for example.
Okay.
So it's non-random mating, basically. It changes things a lot.
Charlene has raised.
Yes, just maybe before you had a question of Roman on the chat, so maybe you can take it before. I don't know if you see it, Diala.
I can't seem to have access to my chat for some reason.
So I can read it. Could the decrease in genome size also be understood as a way to increase the recombination rate, as recombination is less effective with selfing?
That is an excellent question, and I think it might be. But again, we have to. This is where it gets very confusing, and why it was very difficult for me to actually do this. It's not clear if it's the recombination rate that's responsible for decreasing the genome size, or if it's the lower genome size that favors increasing the recombination rate. We don't know. We don't know, and I don't know how to test it. So maybe it's something that happens, like let's pretend that self-fertilization evolves. It starts selecting against individuals that have too much genetic baggage, like too many transposable elements or unnecessary parts of the genome. So it starts selecting for higher recombination simply to slim down the genome, or I'm saying this wrong actually. Again, I'm giving a direction, although there isn't one. And it's complicated. It keeps coming back around and around, basically. So where do I start? Do I start with a smaller genome size? Do I start with the purging process due to selfing? Or does recombination increase first? I don't know. So I don't know if I answered your question, Roman, or if you have another one.
Yes, thanks.
It's not straightforward.
Yeah. And you have also an historical element that you can think of everything at equilibrium. You have to think about what happened.
Exactly. At equilibrium, it's a lot easier to think of it. But this, like, even when we look at the distribution of genome sizes within a given population, we're already not at equilibrium. There's such a large distribution that is linked to either transposable elements, or there are genes that are linked to immune response, or there can be translocations or introgressions or whatever in different individuals. And these can all be maintained simultaneously. And I think it's important to remember that when we say we have the whole genome of a species, what we have is a model of the whole genome of an individual that we're going to use to be able to genotype other individuals. And in that genome, we're not going to have everything. There are a lot of regions that we're not going to have access to because the genes are repeated and we can't have access to that. So there's no easy solution. I don't have an easy solution.
Okay, thanks.
No.
Charlene, you wanted to?
Yeah, I had a question, and I maybe like that you hear better. Yeah, I had a question on mutations. Do you know if there are more mutations on selfing species And if the distribution of mutations is different, because you presented an example where you said that it was difficult to infer distribution and mutation just looking at the generation 17, but do we have more knowledge on the distribution of mutations in, if there are any differences between selfing species and outcrossing species?
So there don't seem to be any, for the time being, it's not clear that there are any statistically speaking. So more like more complete studies have been done now using whole genome data. But once again, the problem is that these are statistical methods to infer the effect of the mean effect of each mutation within the population. And the assumptions that these statistical methods make also rely on some form of equilibrium. So there are some things out there. There doesn't seem to be, with the current statistical methods, there doesn't seem to be a very big difference in the distribution of mutational effects between selfing and non-selfing. As to the mutation rate is extremely complicated to infer. All we have are the mutation accumulation lineages, and they're problematic, like I said, because there is a bias in what you can actually sequence and what you can actually see as a mutation. If the individuals don't survive, you will never sequence them. So you'll always bias your mutation rate in some way. So it's not, I don't think that there is a way around this in any way, to be honest. It's something that we're going to have to accept, that we won't be able to know.
Okay, because it could be also an explanation of why they survive so long.
I mean, it could be. There are several things. It could be the fact that they are able, because of the high selfing rate, which will favor the higher recombination rate and will favor the better purging. It's possible that they're able, through recombination and deletion, to very quickly select for individuals that no longer have parts of the genome that aren't necessary, basically. That's a possibility. That needs to be tested. That needs to be modeled, and it needs to be tested. There are some people now who are trying to develop models to test this. The problem is sometimes the hypotheses that are being made are not biologically plausible. And I don't know how to go around this, to be honest with you. Like, for example, in the robustness case, the hypothesis that you will have gene duplications and that they all work perfectly is too strong of a hypothesis, because it's not necessarily true. What would be the mechanism behind that? And why would they all be maintained functional? And why would they? So we need to find a way to navigate through this. I don't know all the literature, so if there's anyone who's read any of this and has any ideas, please let me know. I would be more than happy to talk about this.
Thank you very much.
Thank you, Shana.
I also wanted to ask, in fact, if this difference between the neutral theory and the selection theory somehow is, in fact, it's a question of time scale, because I would say, in any case, selection acts, but I mean, Darwinian selection is always there to select species. But if it doesn't have time to act, then the neutral theory could be a good approximation. Does that make sense? It's also connected with the mean time to wait between two mutations somehow.
So what you mean is, would it be like the neutral theory would work on a very long time scale, and that the Darwinian selection theory would work on a more day-to-day scale. This is what you mean.
I would say the contrary, in fact.
Okay, I see what you mean. Okay.
Because on the long run, you can select species who are not fit enough. But if you look at a very short time interval, then this selection doesn't have time to enter the game, and so you just observe the drift of the one that you mentioned, that you could have addition of bases, of deletions, but not really due to a misfunctionment of a cell or a species.
That's a very good question. Like intuitively, I would have put it the way I said it, where we would have potentially more selection happening on the short term. And then at the end, we would see patterns that were very close to drift. But the way you're saying it now, it might make more sense to do it this way, to think of it this way. So we would have these changes that are there. And then at some point, one of them goes to fixation. And we go from there, basically. And slowly, there's a selection that happens on a more gradual level. Okay. I don't know.
Because I don't know if there are really good alternatives to selection theories. Somehow it seems to be like the explanation.
Well, I mean, the explanation for that Lynch supported a lot in his book and in several of his papers was really that in any case, like you said, so on the short term, it will also just be drift because it makes very little difference.
Mm-hmm.
But then he also considers that on the long term, the cost necessary to keep decreasing the genome size without without there being a mistake and removing parts of the genome that are necessary would be too high. And so it would only be very light selection just to decrease it when it becomes too expensive metabolically speaking. So this is the nearly neutral theory model that exists a lot. Now, I don't have a very strong opinion either way. No, that's not true. I like to think of the selection aspect more, but I'm not convinced either that I am that it's the only thing that works. So it's really like a balance between the two things happening at the same time, and that is kind of what would make sense. But yeah, on the short term, you're right. It would probably be more of a question of drift because the difference between individuals would not be that big. So drift going slowly up to a certain point, and then it would tip over.
Maybe causes something that makes the individual too bad to live, to produce, and then the effect of selection is removing it somehow.
So it would be kind of like a mutational meltdown, but not like that, basically. More with removals. Okay. It's worth thinking about. I don't know how we'd model it, but it's worth thinking about.
But yeah.
But, sorry?
Maybe we can hear Sylvain's questions before I continue, okay. Because I'm monopolizing the.
Okay.
Sylvain, please.
Yeah, thank you for the question. Yeah, I was wondering whether it would not be worth to have a look at how genome duplication occurs. I mean, I don't know if there is, for example, a bias. If you have in a genome, let's say we have two genes on a genome, and one gene is a bit larger than another one because of an insertion, for example. And what happens during the duplication of the genome and during meiosis? Is there a bias to have some errors into the duplication of the gene to increase the gene which is smaller, or to decrease the gene which is larger or longer? And maybe if there is such a bias, such a molecular bias, then maybe it could explain why we have larger genomes in outcrossing species, because I would expect that in outcrossing species it occurs much more often that that genes with different size occur in the same individual. So maybe it could be worth to have a look at what happens molecularly speaking during gene duplication.
So there's
genome duplication. Yeah.
Thank you. So there are some models that are trying to figure out what happens with gene duplication, and for the time being, they're not explicitly taking into account this difference in molecular size potentially. I don't know what it would do. Do you think that there would be a mechanism that would favor a bias one way or another?
Yeah, we know that there are some molecular bias for recombination, for deletion or insertion, and it depends on many things, on the structure of the DNA. So I would not be surprised if there would be some kind of bias regarding the size of the genes, these kind of things, but I really don't know. It was just a suggestion.
Yeah. Okay, no, I don't know, because I know like gene duplication, not genome, but like genome duplication is one problem, but gene duplication is also a different is a question of interest where they're still trying to figure out what dynamics in general are favored in, for example, maintaining a second copy. Why isn't it removed immediately? Why is the non-functional gene removed? In what cases does it become does it gain a new de novo function? But I will look into that. I hadn't thought of the differences that could be on that level. Thank you. Okay.
In fact, what do you think is the right function that measures the not the genome size evolution, but the relevance of the genome size? Is that somehow the biomass associated to the whole genome of a whole population? So say you are studying a specific species, you spoke about the total number of individuals and also the total genome size of one individual. What is the right function of those two variables, which would be a good one to study the relevance of the genome size somehow?
To be honest, I think we need the distribution. I think we need the distribution of genome sizes in a population, because I think that is when you will be able to see the dynamic changes happening. It's going to be on that level. I think when it's already on the mean level between species, there's already too many differences between the two of them. And so the dynamics might not be based on the same thing. And this is why I was trying to find studies where they really looked at genome sizes, different genome sizes within species, within populations, sorry. But it's so difficult to measure because it requires a different approach. And now what we're mostly doing in population genetics is we have one scaffold, not one scaffold, sorry. We have one reference genome, and then we use that to genotype everyone else. So any changes in genome size, we might not see them, depending on how the reassembly is done, so there are some things that we're missing out on. But again, maybe genome size is not that important. Maybe this is just something that is correlated and it's mostly drift. There's some selection, but it's not that important. And what's important is how many transposable elements do I have? How many coding genes do I have? How much non-coding do I have? And maybe it's more a question of qualitative components and not the actual whole quantity together. But I don't know. what would be best.
Yeah, because somehow it's coming back to like information theory. Somehow it's how much information is compressed.
Yeah.
And why do you have diploids and haploids? Somehow it's why do you have twice the information? It's to reach a certain goal for certain species. So we have to quantify that in some way to take into account.
Like I think especially one of the issues that so diploidy and haploidy, the theory that I've grown up with is that because of the diploid function, or the fact that you're diploid, you can cover a much larger space of the phenotypic space, basically. And this could actually be advantageous. And there was also the other side of this, which was the fact that you have two copies. If ever you have a mutation, it's okay. You have a backup. You have a spare tire, basically. So it's fine. You can live with that.
This, from the point of view of information theory, it makes sense. It can be quantified somehow. But the phenotypic, this I didn't know about. So the phenotypic landscape, somehow you can.
Well, I mean, if you only have one haploid genome, there's only a limited, how can I put this? Because each individual will only have a single copy of a genome, basically, or just a haplotype genome. It can't go as far away as it could if it had two copies that were going in the same direction, for example. Let's say I have an allele that will move me along the gradient of a phenotype. If I have two copies of that allele, let's say it helps me produce a protein, I don't know, if I produce a protein, if I can double the production of protein because I have two copies, it can take me much further. It's kind of in that point of view. It's in that situation. So, but yeah. So it was a lot of information.
Yeah, it's hard to synthesize somehow.
It was difficult for me to bring everything together because it kept going round and round. And
I don't know if you have time for another question or.
Yeah, sure.
Yeah. I just thought about things that maybe it has nothing to see with and to do with all the things you told us today. And maybe it's a very crazy idea, but I know that in psychology there's a law which is called the Weber's law, which says that the error that the human make when they try to copy something is proportional to the size of this thing. So let's imagine that you tell someone that okay, let's try to copy this stick, and you have to be as close as possible to the length of the stick. And in fact, the longer the stick, the higher will be the error made by the people who is trying to copy the stick. And so what's behind this is that it should select for a smaller stick, in fact, because the smaller the stick, the lower will be the error in the length of the stick when you try to copy it. And so there are some archaeological data showing that, in fact, there can be some trend for a decrease in the length of some objects in archaeological data. And maybe it's crazy, but can we imagine in some ways that maybe when you try to copy a genome, is it possible to imagine that the larger the genome, the larger will be the error of the size of the genome that you will make when you try to copy it? And maybe this could be something very similar happening here that tend to make the genome smaller only because of this proportionality between the size of the genome and the error you make when you try to copy a genome. Maybe it's crazy, maybe the analogy is not good, but maybe.
No, I think it would make sense. And I think that's why I'm wondering if that's not why there are also, so this might be crazy. That's not also why there might be more transposable elements in outcrossers that are being maintained partially, because you have the large genome, you copy it, you remove some of them, but then they can come back, and then you remove some of them, then they can come back, and they're maintained at a kind of equilibrium without really being selected against. Because when you have too many transposable elements, which is another problem, you'll end up creating, you can create a huge mess when it comes to recombination, for example. So this might be a very simple, neutral way that is keeping the number of transposable elements down. And it could be worth looking into, actually. I don't know if that makes sense, but
I would say it's what you presented in the drift part, right? This effect that Sylvain is describing.
But in the drift part, the neutral theory was favoring mostly small deletions. And what I understood now was it could also be larger deletions. Or am I wrong?
Well, in fact, the error is not, well, it's really a question of a balance between the thing that you transmit and the exact, I don't know how to say that. When you transmit something, the thing that you transmit is closer to the parenting if the parenting is smaller, because the error you make regarding the length is larger and is proportional to the length of the object. That's basic, but it's a property of the human way to perceive and to perceive stimulus. So I don't know if there is an analogy, because it's a psychological law, you know. But maybe there is something similar, but I'm really not sure about that. But maybe there is something more general than only that goes beyond the human perception.
I really don't think it's crazy, because the error, I mean, some of the mutation you can have on the genome replication, etc., is due to some error of the DNA polymerase. And I mean, it goes for my past classes is a long time ago, but I think the probability of the error of the ADN polymerase is kind of like one base pair to, it makes an error of 1 to 10 power to 8. So as you like have a genome which is large, then of course you would have more probability to make an error, and then you will pass on some mutation to the next generation. So I think it's really, really not crazy.
But also I think like another aspect that will come into that is the fact that it would be, with a larger genome, it would also be more expensive to repair these errors. And I think that's something else that comes into it from that point of view. And so maybe,
Yeah, but I think it could explain a general trend for the evolution of genome size, but it would not explain why you have some coevolution between traits and genome size. For example, why there are some larger genome size within altering species rather than certain species. So there should be something to see with heterozygosity, these kind of things.
But I also think, I'm wondering if it's not, because selfing, what's interesting is that on the one hand, you have these smaller streamlined genomes. And I didn't talk about this today because I think my brain would have exploded. But on the other spectrum, you have all of these polyploids that are either hybrids between two species, and you end up with two whole genomes from two different species, or auto polyploids, where it's an individual, instead of having 2 copies of their genome, they have 4 or 3 or whatever. And selfers are able to maintain polyploidy pretty well as well. And I'm wondering if it's not going to be a bimodal distribution of genome size with selfing. Whereas on the one hand, if you're able to, because if there's any recombination or mutation or whatever, a problem with copying that is removing parts of the genome, when you self-fertilize, if the right parts of the genome were removed so that they don't affect your viability, because of self-fertilization, you can maintain that more easily in the population, basically. And on the other hand, when you have these huge genomes that are polyploid, it's also easier to maintain the status quo, because then you don't have the same issues with recombination that you would have with the outcrop. Wait, would that be it? Yeah, if you're producing it yourself, it's always easier to make sure that you have the right copies that are aligning during recombination. So I think it would be between the two.
In fact, it's linked with this question of what is the right function of the number of individuals and the size of the genome you should consider. Because somehow what Silva mentioned is that, of course, the smaller the genome, then the smaller the probability of making one error, and at least one error. But then it can lead to a high death rate somehow. And if you completely change your the way you produce, then it can completely change also your genome size. And it's like a much different genome size, but for maybe a much different number of individuals also. So it's not clear how we should.
This is why I thought this would be perfect to talk about it here.
Also, something I wanted to mention is that the frequency of mutations can also depend on the life history of some individuals. For example, if it's infected by some virus or something. So depending also not only on its natural death rate or something like that, taking into account several interactions between individuals can modify the mutation rate. So it's not.
But in any case, we know that the mutation rate, we know there is evidence that the mutation rate is actually very variable already between individuals. And also when you take the same genotype, for example, bacteria, and you put it in another environment, it can also have a different mutation rate linked to that. So this is another simplifying hypothesis that we make with regard to the mutation rate. It's not, I mean, even between individuals, it's not going to be fixed. And then even for a same individual, depending on the situation it's in, it might not be the same throughout its life. And we know in any case that for humans, for example, this is one of the classic examples, as we age, we get more mutations. So there's also an aging factor happening. So depending where you are in your life cycle.
Mm-hmm.
So a lot of the mutation rate is but it's another issue.
Yeah, but it's relevant because it's bringing errors and possibly can make individual die faster. Right?
But how important is it? How important is it to have this variance? Does it really bring us enough information, or is the mean enough? I mean, if the mean is enough, great, no problem. But I don't know how to test if the mean is enough.
Mm-hmm. But maybe it could make some link between the two talks. This question of infections and bringing a different mutation rate. But I don't know if Cornelia is still.
Sorry, yeah, I'm here.
Yeah, I I don't know if in your model to make some link with the biology. In your model, could you tell us if you have possible numbers for your the parameters of your model? Do you know, for example, so this beta, so you had this number of edges, the n, which was n to some power beta, and I think biologically it reflects like the number of trials, this stage.
So I thought more. For example, if some bacterium is located in some biofilm or something like this, then in fact they have some connections between the different bacteria, and via these routes also these phages can move to the neighboring bacteria. So so that's something that exists. But yeah, about the parameters, I just was interested in in this effect of this population structure, and wanted to figure out in which parameter regimes this invasion really gets likely in comparison to a complete graph. So this was not really biologically motivated, but
you don't know in which regime
and of course you have always this configuration model, and you always don't have these short loops and things like this. This is of course problematic, but from a mathematical point of view, you can analyze it. You have this coupling with the Gordon-Watson processes.
Sure. But it is to make the link. Is that linked with mutations somehow? So this CRISPR mechanism, does that induce mutations in the cells? Is that
so so there is always some tendency to lose these fragments and also to gain these fragments. You cannot of course, so because it's inherited, you cannot keep everything, so therefore they have to get lost at some time point. It's it's a bit like this genome size. You cannot make it only larger and larger. And they are
the DNA is added to the end of the actual DNA of the cell.
So no, in this CRISPR, this is more on this CRISPR mechanism in the bacterium. This is not related really to the anti-CRISPR phages, but it's more on this CRISPR mechanism, that you have this specific locus in the genome of the bacterium where these DNA fragments of the viruses are collected. And when this bacterium reproduces, then some of these fragments get lost and some of these are kept. And new fragments are collected during the life of the bacterium. So there are models of this, how an equilibrium is obtained.
I have also a question for Cornelia, if I may.
Yeah, sure.
Yeah. So if I am correct, you said that one of the perspectives of your work would be to add a dynamic of the cells that could die or divide, those kind of things, right? So I think it could be a very, very nice idea to do that, but I know it could be very difficult, of course, but I think it could be a very, very nice thing to do, especially because there is a very classical question in evolutionary biology where the question is, under what conditions is it possible for cooperation to evolve and to emerge from a population without cooperation? And on the contrary, if you start with a population of cooperators, to what extent it can resist to an invasion of a cheater. So I think it could be very interesting to do that, especially in the case of viruses, and your model could be very, very interesting to look at this kind of question, I think.
This is when the yeah, so there are this kind of deterministic approximations to this. So if these bacteria are kept in this CRISPR blocked state, and then the cheaters can of course use this, and for this they are deterministic approximations. So, but yeah, well, maybe also one does not really need this stochastic modeling. Of course, on a random graph, it's maybe different. I don't know how it's in.
Yeah, but I was also thinking about cheaters in viruses, because in your case, the viruses are the cooperators, right?
Yeah, but they are phages that don't have this anti-CRISPR.
Yeah, exactly.
Yeah, and but the cooperators, they block the bacterium infect for all, and so the cheaters can.
Yeah, that's why they are cheaters, of course, because but there should be some cost in some way. If there are too many cheaters in the population, then of course it will fail. Being a cheater will fail. So
but I have a question. Would it really be a cheater in that case? Because if in any case the phage cannot invade the cell because it doesn't have the anti-CRISPR mechanism, no, the CRISPR mechanism, sorry. If it doesn't have it, it won't be able to invade anyway. So it's not cheating if someone else takes advantage of it.
Can you repeat so? So we have this CRISPR resistant population, yeah, and then we have this anti-CRISPR phages that block this CRISPR mechanism for a longer time, not only for one generation, but for a longer time. And then phages that don't have this anti-CRISPR, they can use these blocked bacteria for reproduction.
But so wait a minute, but if the bacteria are blocked, that means that there's already another virus reproducing in it, right? This is what this means.
So they kill, so of course these cheaters, then also they kill the bacterium. Yeah, this happens. But it has been blocked before. You need this anti-CRISPR phages to block it. And so this keeps off you. Therefore, you cannot. Having only these cheaters, of course, they won't work.
But I mean, I was thinking maybe it doesn't cost that much anyway to be the initial one, because the advantage that it gives you as the first phage that arrives is worth it. And if another cheater arrives in the same cell, because the reproduction also depends on the number of viruses that have infected the cell, doesn't actually advantage you to have this CRISPR mechanism, or this anti-CRISPR mechanism?
So you mean when you infect together with a cheater bacterium, then the anti-CRISPR nevertheless has half of a chance maybe to reproduce.
So I'm not sure if it's fully cheating in that case. But I don't know. I'm not the best person with this kind of.
No. So if there's a cheater, it cannot block anything. So in this case, the anti-CRISPR would have a very small chance to reproduce still.
Okay.
But it's classical in the sense that if in many cases, if the cheaters are too frequent in the population, then they will not have any advantage. So it's classical in cooperation theory, more or less. And it depends on the structure of the population, of course. And so that could be very interesting to see how the structure of the network could affect the possibility of invasion of a cheetah.
And maybe that is also kept somehow, maybe regionally, or but then you should not have this kind of configuration model where at the second step somehow everything is connected, I think.
Yeah, at some point you mentioned that if you lose the structure of a homogeneous degree, then you are not able to have the same results. But for example, if you consider kind of Erdős-Rényi random graph in the, of course not in the giant component phase, but maybe in the critical one, or it's possible that you have several clusters which resemble what you are studying, right? I didn't think very in details, of course, but it's a way of making the population less homogeneous somehow. I guess in the supercritical phase, it's like in your supercritical phase.
Maybe Cornelia, I can answer this question. So I'm the student who is working with Cornelia on this project, and I think that what we want to do with the configuration model is to say that the critical scaling we need in order to have pairs of edges on the half edges of each vertices is to say that it's something of square root of the number of half edges. And so in the Erdős-Rényi graph, let's say with a probability p for an half edge to, let's say with a probability p to have an edge between two vertices, then the mean number of the neighbors for one vertex is still of order n, like n times p. And so this is a little bit like in the complete graph, where you will need to have square root of n phages in order to allow to see some pairs of edges on your half edges. And so by choosing the Erdős-Rényi graph, there will be no really big difference than in the complete graph.
For p, which doesn't depend on n, I guess. But if you make p
yeah.
on n,
yeah, maybe, yeah.
And take the critical one, then maybe you can do something. I don't know.
Yeah, you're right.
But of course, yes, if there is a giant component, it should be as in your supercritical. I agree.
What kind of components that then will arise if they have very, very, say, heavy tail degree distribution or things like this?
Yeah, I don't really.
So that it cannot spread further or things like this. I haven't really thought about it.
Yeah, I'm not sure about the degree now. Good for what I wanted. I had a question about this, the number of killed hosts that you mentioned in the middle, actually, of your talk. You said you are almost able to prove that it's a Bernoulli. It has a Bernoulli distribution with a parameter p, p Poisson, right? Yeah. But why isn't it a consequence of what you did? Because somehow, at least in the upper bound.
So there we don't know. So far, we don't know how large is the fraction, that is invaded. So with this backward processes, we can only say up to this frequency U will be killed. But in fact, we think that in fact much more hosts get killed, because the larger the number of killed hosts, the stronger is this effect that from different edges a host will be attacked. So the more likely is I can share my screen. Yeah, here it's open. So the larger is this ln, the larger is this rate at which the two hosts will be attacked from different edges. And therefore, the faster it will increase, and yeah, then in fact, at some time point, it will just, I think, more or less in one generation, it will be infected a total proportion of the host population, and then you have another generation where you have U times n many new hosts infected, and they have all Vn phages that are generated. So you have n times Vn many new parasites, and they will. So the probability that any for living host to not to be reached is very small. Mm-hmm. So there will be a few, yeah, maybe. And then it depends on if these parasites, if they hit an empty vertex, if they still live further or if they are killed. Mm-hmm. And if these other hosts also will be killed or not, I think.
Yeah, and maybe last question about your talk is, in fact, biologically, is that really a parasite that co-lives with its host, or could it kill it? By that I mean, could the total number of individuals change or vary in time, for example, decreasing?
This is so the host is, oh God, I think this is so this phage and also the bacterium is very frequent, and it's also it lives, this host lives in very different environments where this anti-CRISPR has been observed.
But it can, I mean, remain in these cells.
So it's non-lytic. That it's this, yeah. That it's non-lytic viral cycles.
Yeah. So cannot.
I don't remember if this is also possible for this kind of phage.
You looked at a specific biological case, you mean?
Yeah. And I don't remember if in the experiments they especially were selecting for these anti-CRISPR phages that really kill their hosts. So they remove this kind of other phases where they just integrate the genome in the bacterial genome, or if it's naturally only the case. I don't remember. Sorry.
Yeah, just that then, if it can be the case, then I would say it naturally this total number of individuals, they should it should decrease in time, right? You should add some probability of each vertex to die out somehow, to be removed from the graph.
You mean so if the phages are literally in the in the bacterium, and then they wake up at some time point and kill the host. Or what do you mean?
So something like that, yes, after an exponential time or something like that.
Yeah, but I think it's more that the bacterium has a chance then to reproduce and to increase in size. Yeah? I would say this is maybe the purpose of not killing the host immediately.
Yeah,
or I don't know.
Make sense.
Yeah, but
you are right.
I don't know.
Just to
yeah.
Yeah, maybe it's more mathematical.
I don't know.
Yeah, it's a way to say what can we do with a varying number of cells, somehow of nodes in your graph.
Yeah. Yeah. So it's so that it's changing over time. Yeah. Yeah, it's I thought about it a bit like, is it that these phages are invading new habitats, in which case this is maybe likely that it happens.
And in this case, you said the DNA is added to the host, right?
It's inserted somehow. Yeah. Inserted. Yeah. And if it's successful, then the host generates this offspring. Yeah. For the viruses, usually they cannot do this by themselves. They need the host to reproduce. But this CRISPR, it's very widespread in very different bacteria. They live very different. So I could imagine that there are phages with only a lytic phage, I could imagine that there's everything around, and also they are very different of these anti-CRISPR mechanisms. They find.
Because somehow it's interesting also from the point of view of Diala's question, because if you think about the phages population, then it's conditioned on the host population living, and so how do you see this genome question in this case? I don't know, because it's not, so the environment is not, independent from the species. It's from
the environment.
Yeah, exactly.
Yeah.
Yeah, I don't know. It's I have to think more.
Yeah, so they have this very fast reproduction cycle, yeah. And I think for viruses, at least those that don't remain, I don't know, you know maybe better, that they don't have some persistent cycles or latent cycles. Their genomes are relatively short, yeah. I would guess. So I know these herpes viruses, they are persistent. They hide in the host, and they have this really large genomes, but they don't have to reproduce also so fast. Yeah. But so these lytic phages, they really need the host machinery, and so I think they should have a tendency for short genomes. So it's really important.
The question is maybe just included in the survival of the host population somehow.
Strategy, yeah.
Well, I'm giving also the right to anybody else to enter the discussion and to. or to drink coffee. Paul?
So maybe I have a question for Diala. So you mentioned looking at genome size between species, and you also cited Lynch several times. And Lynch is, I think he put forward the phylogenetic mixed model a lot in the 90s.
Was it him or was it someone else? Wait a minute. But yes, I know the model. I know the model. Okay, yeah, but I'm trying to remember. Yeah,
maybe it's not the same Lynch.
I think there's only one Michael Lynch who works on these things.
Okay, so that should be him. So I was wondering whether you could apply these models to genome size in order to compare genome size across species, as you mentioned that the time frame is really not the same as the one you were looking at for very short times, and whether it can answer some of the questions we're asking, or whether it's just a completely different.
No, some people have already applied this. I don't know if it was the phylogenetic approach, but they have applied this comparison across species of genome size, and they compared life histories and whatever. But it's often, unfortunately, we always come back to the point where it's inconclusive, because there's so much happening at the same time with life history and with wait, what else was, I think the biggest problem is life history. Actually, it always comes back as a problem. So there are cases where where they find correlations, but we don't know what is causing what. Is the genome evolving first, and it's like pulling the life history with it, or is the life history what's favored and it's pulling the genome with it, or is everything happening at the same time? And I think this for me is the biggest issue, because I don't know, again, I don't know if it's important, but it's something that I think it would be good to say it's not important. It would be good to make a model and say this is not important, so that we can stop asking the question. But yeah, but I don't know if I don't know the method very well. I've just heard of it though, and it would probably be applicable in this case, I think, as a trait.
For instance, at the very beginning, you showed some kind of line with genome size, and I don't remember what was on the x-axis.
The x-axis was genome size, and the y-axis was the recombination rate or the mutation rate.
And so, for instance, for this regression line, did you apply phylogenetic correlation?
I don't remember. It's in Lynch's paper. The book is here. Let me tell you, I think they did not account for the phylogeny because there wasn't enough data. So this is also another problem. It's like we don't have enough data. No, so it's not like there are different regression lines that are drawn depending on the group of species. So for example, they separated the unicellular from the eukaryote species, because there's clearly a different tendency that's happening. But other than that, there was no control for the phylogeny. And at the same time, I don't know how much of a role the phylogeny will actually be playing on this level. Because if the genome size evolves too fast, you might not see it might not play a role.
But you might have some inertia effect where you
might have yeah, of course. I think it will depend on how far in the past the species diverged.
Because on this curve you showed, there were clearly some groups in different colors. And
yeah, that's
it looked like groups were doing the trend, and inside of each group it looked like the trend was not that clear. So that's why I was wondering.
Yeah, I think the trend in general it's not as strong when you look at each group separately. But again, we have to be careful, because is it depending on what was genotyped? Was there a bias in what we selected because of life history? Was there a bias in what we selected because of environmental accessibility? Was there? So I think that also needs to be. We need more data. Actually, we need more information. But the regression normally, the correlation is still there.
Okay.
Let me look at the figure actually. Yes, so let me, if you're interested in seeing, so basically what you can see here, so these are the unicellular, which are alone, and this regression is done only for them, basically, this part here. And then here, like you see clearly all the plants, they're going from here to here. So they do follow the correlation. The vertebrates are a bit more grouped up, but that's also because there's less variance in the vertebrates when it comes to genome size. And that isn't clear why. There seems to be something happening that's constraining it. And then the last one is the invertebrates. You're right as well, the invertebrates don't have the same correlation.
Do you know what vertebrates is to the right, the rightmost?
You mean the one that's
biggest,
all the way? Okay, that one
the largest.
I have no idea what that would be. Like I can look at the paper and see what it is. Which one it is.
Interesting, because it's much larger than all the rest.
It might be humans.
Might be humans.
Wait, I have the paper, I can look at it. But yeah, but if it's not that, I can send you messages.
Mm-hmm.
So there's a lot of but is it important?
No.
No, I mean, it might all be phages, you know. It might all be insertions from viruses that we've gotten.
Well, do you have other questions, Paul?
No, thank you.
Yeah. So I think maybe we can stop here, and thank you very much again, Cornelia and Diala.
Thank you.
Those very nice talks. And if you can send me your slides, it would be very nice. You can put them on the website. And